The mouse, this tiny creature, has had a huge impact on science research. Their short life cycle, small size, and genetic adaptability make them ideal for studying genetics, disease progression, and development, enabling scientists to model human illnesses and test therapies to better understand brain function. But why are mice so useful in brain research? What makes their brains so comparable to ours, and where do the similarities and differences lie? This is precisely what we aim to explore in this short video, namely, in how far a mouse brain is comparable to a human brain. So the most obvious difference between the mouse and human brain is size. The human brain weighs approximately 1,300 grams, while the mouse brain weighs about zero point 0.4 grams. The human brain is over 1,000 times larger than the mouse brain, with approximately 86 billion neurons and countless interconnections. But studying it directly is incredibly challenging. That's where the mouse, with only approximately 71 million neurons, comes in. Mouse brains are simpler and easier to study than human brains. Humans and mice share common ancestor from approximately 80 million years ago, leading to significant genetic, structural, and functional parallels in brain. Let us first look at the structural and functional parallels and divergences. So the cerebrum in humans is exceptionally large, occupying about 75% of the brain's volume, and is highly folded with gyri and sulci to maximize surface area. In mice, however, the cerebrum is much smaller, smooth, and constitutes only 30% of their brain. In contrast, the cerebellum, responsible for motor coordination and balance, takes up about 10% of brain volume in both species and features Purkinje cells and similar microcircuitry. You can see these Purkinje cells of the mouse and human cerebellum in this picture as well. So these similarities in cerebellar structure allow scientists to use mouse models to study cerebellar dysfunctions, such as ataxias, which lead to movement disorders in humans. When it comes to sensory processing, both mouse and human brains use analogous neural circuits to process sensory information. For instance, the visual, auditory, and somatosensory cortices in both species are organized into distinct layers and columns, enabling parallel processing of sensory inputs. However, the reliance on specific sensory modalities differs. Mice rely more heavily on olfactory cues for navigation and social interaction, whereas humans rely predominantly on visual and auditory inputs. This distinction is even evident in the relative brain proportions. See, the olfactory bulb is relatively larger in mice than in humans. As you can see in the picture, both human and mouse brains allocate significant volume to the cerebral cortex, which is responsible for higher-order functions like cognition and sensory processing. However, the human prefrontal cortex is larger and more specialized. It is crucial for abstract reasoning, decision-making, and social behaviors in humans. Mice, though valuable models, lack features like a well-developed granular layer 4 in the prefrontal cortex, which limits direct comparisons and the translation of research findings. Now, you might have noticed that mice excel at tasks like navigating mazes and remembering patterns, right? They also exhibit complex behaviors like learning, memory, and problem solving. However, these behaviors rely on conserved neural circuits like hippocampus, which, by the way, works remarkably like ours. It plays a critical role in spatial navigation and memory formation in both species, with homologous structures including CAON, CA3, and dentate gyrus regions, and molecular pathways. As a result, Alzheimer's research, which focuses on hippocampal degeneration, can be done using mices to study amyloid plaques, tau tangles, and memory loss progression. Similarly, structures like the basal ganglia and amygdala are conserved across both species. And the shared conserved neurotransmitter systems in mouses and human, like dopamine and GABA in the basal ganglia, helps the scientist to study dopamine loss in the basal ganglia and its effects on movement in Parkinson's disease. Does that sound exciting? We have talked a lot about the basic structural and functional comparison. Now let's talk about the genetic organization. Well, it is quite interesting to note that despite the size difference, mice and humans share about 85% of their protein coding genes, which also highlights their evolutionary closeness and makes them a powerful model for understanding brain function and diseases. Did you know that over 99% of mouse genes have homologous genes in humans? Genes like PAX6, critical for brain development, particularly in eye and forebrain structures, and glutone, essential for brain energy metabolism, are highly conserved between the two species. This homology with the human genome enables the modeling of various brain functions and disorders. In fact, 90% of disease-associated genes are identical in mice and humans, making mice invaluable for investigating brain diseases. An example of how the genetic similarity between mice and humans supports their role in brain disease research is the APP gene, which is associated with Alzheimer's disease. So, a lot of this might be associated with Alzheimer's disease. In both species, mutations in the APP gene lead to the accumulation of amyloid plaques, a hallmark of Alzheimer's. This genetic similarity enables researchers to use mouse models to study the progression of Alzheimer's and test potential therapies. Interesting, right? Now, despite genetic and structural similarities, there are limitations to using mice in modeling human brain function, mostly due to the absence of human-specific genes. For example, the lack of human-specific genes like RHRGAP11B, which drives cortical expansion, limits the applicability of mouse studies to higher cognitive functions. Moreover, the absence of genes like SRGAP2C, which contributes to human brain development, cognition, and neural connectivity, makes it difficult to study psychiatric disorders like schizophrenia, which rely heavily on these advanced neural networks and human-specific brain traits for their development and progression. Now that we've explored the genetic similarities that form the foundation of mouse and human brain studies, let's dive deeper into the cellular level, where the organization and function of neurons and glial cells reveal even more about their shared and unique roles in brain activity. At the cellular level, both mouse and human brains share basic organizational principles, using similar mechanisms for action potential generation and synaptic transmission. Both mouse and human brains have similar neuronal types, such as pyramidal neurons and interneurons in their cortical layers 1 to 4, though the structure of these neurons differs. Example of this would be pyramidal neurons in the temporal cortex, with human neurons having longer and more complex dendritic structures. Furthermore, another interesting research has shown that both human and mouse brains share similar inhibitory circuits, which are made up of excitatory pyramidal cells and inhibitory interneurons, P-valve and STE cells. But how is this similarity relatable for the research? Well, it is related because the balance of excitation and inhibition regulated by these circuits is crucial for conditions like autism and epilepsy. Thanks to the similarities between these inhibitory circuits, we can use mice to better understand human neurophysiology, ultimately leading to develop improved treatments for these neurological disorders like autism and epilepsy in humans. Now, we know that mouse and human both have glial cells. However, astrocytes, one type of a glial cell, work differently in humans and mice. Studies show that mouse astrocytes are better at handling oxidative stress, a key factor in many neurological disorders. In low oxygen conditions, mouse astrocytes kick into repair mode. Human astrocytes, not so much. On the flip side, human astrocytes are more active during inflammation, something we don't see in mice. These differences might explain why more than 90% of drugs for brain diseases that work in mice fail in humans. To address this, scientists suggest modifying mice models to better mimic human astrocyte behavior, particularly for studying diseases like Alzheimer's and strokes. Now that we've covered key aspects like the structural, functional, and genetic comparisons between mice and humans, let's circle back to our original question. So how far is a mouse brain comparable to a human brain? Quite far, actually, both in basic shared brain structures like the hippocampus and neurotransmitter systems, and in conserved genetic pathways. These similarities make mice invaluable for understanding conditions like Alzheimer's, Parkinson's, and epilepsy. However, it's also important to recognize the differences that persist. While mice and humans share basic brain structures, humans have more complex regions, particularly in the cortex. Functionally, mice exhibit fundamental brain processes, but their cognition and brain development differ significantly from humans. Additionally, cellular differences, like the behavior of astrocytes, highlight the limitations of using mice as perfect models. Remind us that mice are only a model, not a perfect replica. But it's important to remember that what we covered today is just the tip of the iceberg. There's so much more to explore about how mouse brains both mirror and differ from ours. That's why scientists are constantly refining mouse models to make them more human-like, helping us uncover deeper insights into the complexities of the human brain and advancing treatments for neurological disorders. With this, I would like to thank you for your attention.